Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this connective tissue lecture series, I will be going over fibronectin and laminin. Let's get into the details. Fibronectin is a major glycoprotein present in extracellular matrix. It contains three types of repeating motifs, that is type 1, 2 and 3. And it is coded by a large gene and this gene, when it undergoes the transcription process to make an mRNA for fibronectin, there will be many splicing going on to make a fibronectin molecule. Now the fibronectin in the extracellular matrix, it will interact with different proteins and glycosaminoglycans, and they are heparin, fibrin, collagen and also cell surfaces. So fibronectin, it binds to cells via integrin class of transmembrane protein, as it is shown in the figure here. As you see, integrin receptor, which is a transmembrane protein being part of a membrane there, it is going to interact with the collagen through fibronectin and heparin, which are interacting with integrin receptor, and integrin receptor on the other side of the cell, inside the cell, cytoplasm there. So it is going to interact with 𝐶𝐿in, vinculin and other proteins which are further interacting with actin in a skeletal muscle. Now the sequence of amino acids that are present in fibronectin that will interact with the integrin receptor, they are in arginine glycine aspartate sequence, which is referred as RGD sequence. Remember that. So the RGD sequence is the one, that part of the fibronectin is the one that will interact with integrin. So indirectly, fibronectin, it is going to make a communication with the intracellular components. That's how it communicates intracellularly, and also it communicates extracellularly by interacting with collagen and other collagen associated proteins. Now the collagen, fibronectin, laminin, all are major extracellular matrix proteins that will interact with the cell surface receptors. The interaction of fibronectin with its receptors provides one route whereby the outside the cell can communicate with the inside. Fibronectin is also involved in cell migration. It provides a binding site for cells and thus helps them to steer their way through the extracellular matrix. So these are some of the functions of fibronectin which is being present in the extracellular matrix. Coming to another extracellular matrix protein, that is laminin. Laminin is a major component of basal laminas, and the basal laminas are specialized areas of extracellular matrix that surround epithelial and some other cells. Example, we see that in muscles. As you see here in the figure, so the basal lamina, which is towards the interior of the cell there, the basal lamina which contains the laminin protein, and thus laminin, it's going to interact with type 4 collagen and other associated collagen associated proteins like integrin, destroglycan, N-tactin, 𝗫𝐴𝐴𝐿. So all these proteins which are forming an interaction with the type 4 collagen molecule, and this type 4 collagen molecule further interacting with the laminin, and that interact with the basal lamina. So in basal laminas, laminin forms a network with N-tactin, heparan sulfate proteoglycan, 𝗫𝐴𝐴𝐿, which are attached to type 4 collagen molecules. Now let's look at a little bit about structure of basal lamina and glomerular filtration. So the basal lamina, it regulates the passage of large molecules like most of the plasma proteins across the glomerulus into renal tubule. It allows molecules up to 8 nanometer to pass through. Though albumin is smaller than 8 nanometer, but it is prevented from passing through easily by the negative charges of heparan sulfate and sialic acid glycoproteins that are present in the basal lamina. That's why albumin is not filtered, that's why it's lost from the body in the urine is prevented. When the normal structure of glomerulus is damaged, it leads to alteration in the pore size and negative charges, allowing albumin and other proteins to pass through into the urine and thereby loss of proteins from our body. This is what is the role of fibronectin and laminin in the extracellular matrix of our cells. Thank you.